Hi, my name is Fatemeh Malekipour. I'm a postdoctoral research fellow at the University of Melbourne, Australia. I'm a mechanical engineer focusing on investigation of the biomechanics of the subchondral bone. I'm also an editorial board member of HubLE, and I'm here today to interview Dr. Haniyeh Hemmatian on behalf of HubLE. She has presented her work at ECTS 2021. Thanks, Haniyeh, for joining us today. And could you please introduce yourself to the HubLE audience?
Hello, Fatemeh and everyone, and thank you for having me. My name is Haniyeh Hemmatian. I am a postdoc in biomedical engineering, working at the Department of Osteology and Biomechanics from the University Medical Center Hamburg from Germany. I'm doing research in the field of bone biomechanics and musculoskeletal medicine, and specifically my research interests have been focused on 3D visualization of bone microstructure, including osteocyte networks and how changes in its morphology can affect bone mechanoresponsiveness as seen in bone disease. Particularly using high-resolution imaging techniques like microcomputed tomography, electron microscopy, and also computational modeling.
Awesome, that's great. So did you want to tell us what you presented at the conference?
Yes, well, I would like to take this opportunity to talk about one of our recent projects, which has been done in collaboration with our collaborators from Dresden University of Technology from Germany, investigating the role of osteocyte in osteotropic cancer using a mouse model of bone metastases, which is also, yes, the result has been presented at ECTS conference. And but let me start by mentioning our motivation, which is defined by the fact that both breast and prostate cancer are really common in between women and men, and the 5 years survival rate of these cancers is significantly reduced upon metastasis formation. And actually bone gives a perfect soil to cancer seeds, and patients with bone metastases usually suffer from severe pain and also bone fractures. And even metastasis is the primary cause of cancer mortality. So actually knowing that the key to a successful treatment strategy depends on our understanding of what controls the tissue function in health and disease, we would like to investigate something novel with our tools. And in our research group, our focus is on bone quality, and specifically in this project, we looked at osteocyte, the bone mechanosensory cells, to see how its morphology and its functionality are affected by skeletal metastases. If I would like to briefly explain this project, actually to mimic an established bone metastasis, we used an intratibial injection mouse model of E0771 breast cancer cells and also RM1 prostate cancer cells in order to bring tumor cells into a direct connection with the osteocytes. Thanks to our collaborators from Dresden to provide such models. Actually what we have done was that we used high-resolution microcomputed tomography together with the histological techniques, and we showed a unique osteocyte phenotype, which is linked to either osteosclerotic or osteolytic lesions upon metastasis formation. And in both models, we found that the size of osteocyte lacuna were larger, and also the largest osteocyte lacuna accumulated next to osteosclerotic lesions where we have new bone formation. While at the same time the osteolytic lesions where we have bone resorption presented with an absence of large lacuna. And additionally, using histological techniques, we determined that both newly embedded osteocytes as well as osteolytic osteocytes contribute to this phenotype. And furthermore, we detected a significantly reduced in osteocyte connectivity, and also we found a vast amount of empty lacuna in actually in the regions that we have osteosclerotic and osteolytic regions, and in the presence of tumor cells in both cancer types, which might indicate that the function of osteocytes and the mechanosensation by these cells may be impaired in such areas. And
so basically,
yes.
you're using this as an indication of presence of, what is it, the lesions or the metas, right?
Yes, okay. Right. And actually, these observations may suggest that the local characteristics of osteocyte networks is influenced by the presence of tumor cells in bone microenvironments. But of course, future studies or further studies may actually address or explain how these abnormalities in bone tissue affected by tumor cells can affect osteocyte signaling and subsequently bone mechanotransduction in osteotrophic cancer.
Awesome. So what would you say is the limitation, or basically in a specific way, how far do we have to implement this sort of technique to human beings? Is it like very far, or is it translating easily, or?
Yes, actually, if I would like to talk about the implications of these findings and about the big picture behind these findings. Actually, I would say that, of course, a better understanding of the interaction between the osteocyte and tumor cells may provide, for example, the diagnosis or treatment strategies, for example, to delay bone metastasis development and formation, or for example, to prevent bone destruction, and might be of interest, for example, for the drug developers, and hopefully eventually benefit the population suffering from osteotrophic cancers.
Awesome, great work. So what is next? When are you publishing this work, or have you already published?
Actually, there is some project-related publication, and of one paper. It's currently under review at Bone Journal, and
awesome. Okay, great.
Abstract has been presented at ECTS recently.
Beautiful. So, what is next for you in terms of your career and jobs?
Yes, actually, it's a good question. And in the long term, I would like to pursue a career in academia, and of course, in the field of bone biomechanics and mechanobiology. And actually, the opportunity to have a 2-year HubLE Research Fellowship from Germany already has given me the chance to exchange knowledge and obtain some new skills. But considering my career goal to become hopefully an independent scientist in the near future, I think I will go for another postdoc position and to further improve my current skills, and because I believe that a training in, for example, project management or mentoring the students, teaching and writing grants, might help me to develop the skills that I might need them in future to successfully lead my own research group.
Absolutely. Yeah, sure. Awesome.
I do believe that this might help me to build up my skills. And
yeah, sure. Great. Thank you very much. Great work. And it's got a very nice implication. And we wish you a very successful career ahead. Thank you, Haniyeh.
Thank you, Fatemeh.